have a lot of people, they know malaria only on papers, but behind this every single number, you have a lot of emotional suffering. And if there is really good solution that can help us to end this suffering, they have to help us.
Malaria is a terrible disease. It is ancient, it's been with humans since the beginning of our time. Malaria is affecting mostly the poorest people on Earth and mostly children under the age of 5. And we see more than 400,000 deaths every year. This is a crisis, and a crisis that we need to address now.
The malaria parasite is spread by mosquitoes. Over the last few decades, there's been a reduction in malaria rates, but in 2019 there were still 229 million cases and 409,000 deaths. The current World Health Organization recommended treatment for the majority of cases as artemisinin-based combination therapy. Other tools are insecticide spraying or bed nets, but more help is on the way. There are 3,500 species of mosquito, and around 30 to 40 of those transmit malaria. But, and this is important, it's only the females. Here at Imperial College London, scientists have been working on a radical idea. What if you genetically engineered those females? I've arrived just in time for lunch with Ioana Morionu and friends. So pleased they're in the cage.
Yeah.
And what's on the menu today?
It is cow blood.
Oh, lovely. Is it always cow blood?
Yeah, that's what we feed the females on when we want them to lay eggs.
And of course it's the females that are the baddies, aren't they? They're the ones that bite you. They're the ones that spread malaria.
Exactly.
What's that little gadget there?
So here I've got the blood I've just put in, you can see. And I've attached it a membrane which essentially tries to imitate the feel of skin. So it's made of collagen and other proteins. And when the female mosquito pierces it in order to draw the blood, it's gonna imitate the feeling of like piercing into a human.
Our skin, our skin.
Yeah. And this blood is gonna get warmed up. I'm gonna attach it to this device to mimic the human temperature.
So are there males and females in the cage?
Yeah, but you will see in a bit that the males will be completely unbothered about the blood. And it's only the females that are gonna get attracted to it.
It's probably a bit late to warn you, but this film is going to make you feel very itchy.
So slowly the females are understanding that there's blood there.
This feels very familiar. It's like being at a barbecue. This rather bloodthirsty research is supported by Target Malaria, a not-for-profit international research consortium that aims to co-develop and share novel genetic technologies to help control malaria in Africa. The Imperial research team is led by Dr. Andrew Hammond, a molecular biologist who is currently in Baltimore, USA. Scientists don't usually like to talk up their research, but Andrew is very excited.
It will be revolutionary. Gene drive is potentially a game changer, and we can get complete eradication of malaria. A gene drive is essentially just a very special type of genetic modification. It's something that can copy from one chromosome to another, and that means that instead of being inherited by half of the offspring, like a normal gene would do, it can get inherited by all or almost all of the offspring. And that results in something mind-blowing. It allows your genetic modification, your gene drive, to spread to an entire population of insects in just a couple of years. And you can do that because the gene drive is doubling every generation. Every generation you go from not just half, but all of the offspring carrying it.
There are 3,500 species of mosquito in the world, but the team in London are working with only 3: Anopheles gambiae, Anopheles colutsiae, and Anopheles arabiensis. These are responsible for 95% of malaria in Africa. So how do they actually transmit malaria?
So when someone has a malaria infection, the parasite can run in their bloodstream. When the female comes to feed on them, it picks up the human blood and the parasite through her proboscis. And then it takes about 1 to 2 weeks for the parasite to develop inside the mosquito, and it reaches the female salivary glands. So whenever they bite again, and feed on human blood again, they can inject the parasite from their saliva into the human host bloodstream. And then that cycle repeats and repeats, and that's how you get transmission of malaria.
I did notice actually that you guys are ready for any escapees. So
Yeah.
maybe I should just stand here and just be ready.
Yeah, exactly. We use that to kill any free flyers.
Because I mean, what happens if they do escape? Because I mean, some of them have got malaria, haven't they?
No, actually, we only keep mosquitoes in the lab that don't carry any parasite or virus, so they're completely safe.
Good to know. But I am ready. The gene drive is injected into the eggs, and the breeding process happens here in the insectary.
There's 2 doors to the facility, which both have an air curtain, and the timer on it is to give enough time to blow any mosquitoes away that might be sitting on the other side of the door.
So they can't escape.
So they can't escape from the facility.
It's environmentally controlled between 26 and 28 degrees. It feels like being in the tropics with a lot of unwelcome guests that may bite you. So what do the eggs actually look like? Can you see them?
Yeah, we've got some eggs here. When they're first laid, they're white, and then they melanize over about a 45-minute hour process. So they darken and the outer shell hardens, and they look like that.
Each little rod is an egg, including the sides of triangles, where eggs have clumped together in the fluid. Each female lays on average 120 to 150 eggs. And at what point do you inject them?
So you want to inject them just as they're starting to darken a little bit. So the shell's still hard enough, or soft enough, I should say, to actually get the needle in, but it's not so soft that you'll actually damage what's inside.
But to understand what's in the injection and how it works, Joanna needs to give me a quick molecular biology lesson, if there is such a thing.
Gene drive allows us to manipulate inheritance in a way as to increase the frequency of any genetic modification that we want into population and actually modify the whole population.
Mendelian inheritance refers to patterns of inheritance that are characteristic of organisms that reproduce sexually, where genes are not always passed on to descendants. But with gene drive inheritance, the modified gene spreads not by improving the reproduction fitness of the organism, but by increasing the odds they themselves will be inherited.
Like humans, mosquitoes have 2 copies of each gene, and whatever genetic modification they carry, when they mate with a wild-type individual, that modification would get halved in their progeny pool. So half of it will inherit it and half of it will not. What happens with gene drive inheritance is instead of half and half getting the genetic modification, we have the whole of the progeny pool getting that genetic modification. So at every generation, even if the mosquitoes mate with wild-type mosquitoes, that modification gets more and more frequent in the population. And you can see that it increases with an exponential frequency.
The idea of gene drives has been around for years, but scientists didn't have the technology to do what they needed to do with the organism's genome or genetic instructions. Then the gene engineering tool CRISPR-Cas9 was discovered, which can be used to change the DNA of animals, plants, and microorganisms with extremely high precision. About 3 billion years ago, bacteria evolved a defense mechanism to fight infection by viruses. When they survive a viral attack, they store a part of the virus's DNA in their own genetic code in a DNA archive known as clustered, regularly interspersed, short palindromic repeats, commonly known as CRISPR. When the virus encounters the bacterium again, it quickly makes an RNA copy from its CRISPR archive and passes it to a protein called Cas9. Cas9 scans all genetic material from an attacking virus until it finds a 100% match and binds to that DNA sequence. The protein then acts like a pair of molecular scissors which cut the viral DNA or RNA. This disrupts the function of that gene and therefore the virus's life cycle. Researchers discovered that CRISPR-Cas9 could also be programmed in the lab to cut DNA sequences of their choosing.
Now, what we do with the gene drive is that we incorporate that editing capability and we give it into the mosquito, so the mosquito can do its own editing. that's the crucial thing. A gene drive is unlike other genetic modifications because rather than using CRISPR to make the modification, we engineer mosquitoes to make their own CRISPR, so they can edit their own DNA in a way that we program.
And what is it exactly that you're targeting in the mosquito? Is it a particular gene?
So we program our mosquito to cut and edit a gene called double sex. And this gene is absolutely essential for females to develop. So if you mutate this gene, male mosquitoes are fine. But the females will be unable to develop, unable to bite, and unable to reproduce. And so as this spreads through the population, you start to produce more and more females that are unable to have offspring, and the population crashes. That is the trick. That's exactly what we're doing.
So what happens when you put the gene drive into the mosquito?
So, like humans, mosquitoes have 2 chromosomes and 2 copies of each gene. Here you can see an illustration of one of the chromosomes that has the target gene in yellow, and one of the chromosomes that has the target gene interrupted by a gene drive in red. And what will happen is our gene drive expresses 2 things that are important to us. Cas9, which is a protein that can cut DNA, and a guide RNA, which is a short sequence that's complementary to our target and will lead the Cas9 to cut the DNA. So what happens is our gene drive gets expressed and recognizes the bit in yellow, and it cleaves it.
So it cleaves it, it cuts it in half.
It cuts it in half, that's right. And then the DNA repair machinery of the mosquito actually recognizes that these 2 chromosomes are a copy of each other, and this one has a cut, and the mosquito machinery thinks that it's missing something. So what it does is it's trying to repair the cut of the DNA by looking at the other chromosome that has the gene drive and copying off it.
Wow.
And that's how, instead of having 1 copy of the gene drive in the mosquito that carries it, we now have 2 copies of the gene drive. So all of its progeny will inherit the gene drive.
So now it's injection time. Next door to the insectary is where the eggs are injected. Dario Miachi is one of a few with the skill set. It's so difficult, even the super talented only have a 10% success rate, which is why so many eggs need to be injected. So Dario, this is your domain?
Yes.
All right, so what happens now in the process?
So now Matt is gonna align the eggs. It's very important for us to have the eggs aligned so that I can inject always the same spot.
Under the microscope, the eggs are a bean shape, and Dario will inject the pointy end, which is where the sperm and the eggs are formed.
Load. A tiny quantity of DNA, usually 1 microliters.
Oof.
Yeah.
You have a steady hand, Dario.
Yeah. I mean, you get there with practice.
I'll leave it to you.
And then we can load the needle into our machine.
The first generation will not have the modified gene, but after that it will be passed on to almost all descendants, and that will make the females infertile. They also include fluorescent protein in the injection, which shows them if the gene has been successfully passed on. And they can see this when the eggs hatch into larvae.
Matt, can you turn the lights on? You can see fluorescence in the eyes and in the spinal cord.
Okay.
So the protein that we use here is a RFP, red fluorescent protein, and sequence of DNA that tells the organism where to express the protein and when to express the protein. The promoter is active in the nerve cells, so eyes and spinal cord.
Oh, and there's another successful one.
Yeah.
Fantastic.
I mean, we were pretty good this time.
Well done, team. The next step was to trial it in much larger cages, and they simulated the environment of Africa in a high-security lab in Terni, Italy.
So we might make a strain which performs very well in the lovely environment we've created for it in London. But if we were to release it into Africa, where it's got to compete with the wild, strong mosquitoes, and it's got to avoid predators, and it's got to find those swarms of mosquitoes mating, it might fail to do that.
The results were far beyond what was expected.
We were able to completely eliminate this simulated population of mosquitoes in Terni in Italy in less than 1 year.
Have you trialled this in the wild yet?
No, so no gene drive has ever been tested in the wild, and that's not something that's going to happen in the next couple of years.
But while scientists from institutes and universities from Africa, Europe and North America work away on the technology, another team from Target Malaria has already started fieldwork. Dr. Abdullah Diabate is the principal investigator in Burkina Faso.
First of all, I think the most important thing for us is just to make sure that the gene drive construct in the field will be able, to operate as we really expect it, to operate. We want just to make sure also that this technology can be used for the best of interests of Africa, but in a very, very safe way, for our different populations. And then obviously also there is a stakeholder engagement, which is very important, that we need to make sure that with all the stakeholders in the field, there is a very good understanding of this technology, and then we will have green light from them, to do the work.
In July 2019, Dr. Diabate and his team released around 6,500 genetically engineered sterile male mosquitoes. Their spread and survival was monitored, and the data will be used to inform an eventual release of gene drive mosquitoes.
This is the first time a genetically engineered animal has ever been imported and released in an African country. And it's something that's essential if we're to go from something so simple as engineering a small edit of DNA in a laboratory in London to modifying entire populations of the mosquito in Africa.
And when you hope to release gene drive mosquitoes, how many would you actually need to make an impact?
You can release just a handful. I'm talking hundreds, maybe a couple of thousand, and that can have an impact across an entire region, several countries in Africa. But the reality of it is, what you're likely to see is something in the range of tens of thousands of mosquitoes, not millions, being released in a few locations to give it every chance of success.
So what's the ethics of wiping out the population of a species?
So something we won't see is an eradication of a species, nor is this something that we actually think is desirable. There are more than 3,500 species of mosquito worldwide, but very few transmit disease to humans. Using gene drive technology, we can specifically reduce the number of mosquitoes belonging to the 3 or 4 species that transmit the vast majority of malaria in Africa. And in areas where you see that those species have been reduced in number, you'll see a shift towards mosquitoes that, for the most part, cannot transmit disease to humans.
Ecological studies have been carried out that show reducing or even eliminating the Anopheles mosquito is unlikely to have a significant environmental effect. But is eradicating a species, no matter how deadly, the ethical thing to do? Shouldn't scientists just find another solution?
You go to any other country here around Burkina Faso, in the health center, during the rainy season, you will see a lot of kids laying down on the floor. They are sick, and a lot of them, unfortunately, are dying. And if you see this kind of thing really, and then you have this kind of technology, I think it's really kind of call to your conscience that, well, we have something here, it's new, so we really need, to see how we can use it, for the best of the community, but also to make sure that it can be used in the safe way.
As well as mosquitoes, researchers are working on other animal gene drives to eliminate pests on crops or invasive species. However, there are still a lot of unknowns about gene editing technology, and as they get closer to being a reality, these ethical questions need to be discussed, and regulatory pathways must be put in place to look at each on a case-by-case basis.
We need to get the green light from the different community, otherwise we'll never be able to deploy it, and then malaria will still be killing, millions of people every single year.
I don't think gene drives are the silver bullet that will do the entire job, but I think they are the thing that will push us to the point where we can eradicate malaria.
With malaria increasingly becoming resistant to key treatments and mosquitoes to insecticides, gene drives could play an important role in combating the disease. Target Malaria hopes to release the gene drive mosquitoes in 5 to 10 years, once the staged testing and approvals are complete.
The mosquitoes will spread that technology everywhere that is needed. It will spread it into remote villages where people are affected by disease but do not have access to medicine or the bed nets that could save their lives. That's why gene drives are such a revolutionary tool, such a game changer in the fight to eradicate malaria.
You have a lot of people that are outside of Africa. They know malaria only on papers. They see, numbers, but behind this, every single number, you have a lot of emotional suffering of the parents here in the field. And if there is really good solution that can help us, to end this suffering, they have to help us, to achieve it.
Thanks so much for watching. Don't forget to like, comment and subscribe and hit the bell button below for notifications. We'll see you next time.